Hello, everybody. Good afternoon. Welcome again at the 5th Symposium of Physiology and Pathology of Neuroglia. This is also the institutional seminar of the Instituto de Neurobiología. I have the great pleasure of introduce our next speaker, Dr. Suzana Herculano Houzel. She's a biologist and neuroscientist at Vanderbilt University, where she is associate professor in the Department of Psychology and in the Vanderbilt Brain Institute. She has published more than 100 research articles with almost 20,000 citations. Since 2 years ago, she is the first female editor-in-chief of the Journal of Comparative Neurology, the main scientific publication in her field. Also, since 2 decades ago, Professor Herculano Houzel developed her own method to turn brains into a soup and count cells and proceed to apply that to the brains of more than 100 vertebrate species in collaboration with several researchers around the world, creating a formidable data set that she continues to explore in relation to brain scaling and diversity in evolution. Most recently, she showed that theropod dinosaurs had primate-like number of neurons, that mammalian brains must use what energy they get, not as much as they demand, and the lifespan scales not with body size or metabolic rate, but with number of cortical neurons. Professor Herculano Houzel spoke at TED Global once on the topic, what makes
Thank you, Abraham, for the kind introduction. I want to thank the organizers for the kind invitation, and Monica, you're such a gracious host. Oh, but should I turn off one of these? All right. It's a pleasure to come speak to you on, I don't know if this is, it's a pleasure to come speak to you on what different brains are made of, which is my favorite topic. This is not at all what I trained to do. At one point I was supposed to be a geneticist, then I was supposed to be a visual systems neurophysiologist, then I was actually science communicator working in the Museum of Life in Rio, Brazil, 22 years ago, 25 years ago, when I realized that there was something very fundamental about brains that we actually didn't realize. So the first thing I did when I started working at this museum and I was in charge of developing hands-on activities for the public about the brain was, well, what do people know about the brain, or what do people think they know about the brain but actually get completely wrong? So I prepared a 95 short sentence survey that I asked members of the public to respond, but I also asked my colleagues, senior scientists, senior authors of major publications in neuroscience to answer. And you see here that this was my control group, right? So 97% of neuroscientists sampled believed that we use our brain 24 hours a day, yes. 91% agreed that the mind is a product of the working of the brain. Also, that's why we do neuroscience, right? And you see that a majority, although not even 70% of the senior neuroscientists disagreed with this statement that we only use 10% of our brain. But then, when you look at the responses from the general public, so we see good things, like almost 90% of the people with a high school degree or a college degree or a graduate degree believed that, yes, we use our brain 24 hours a day. That's where our mind comes from. And you see there that essentially 60% of people in Rio with a college degree believed that they only ever used 10% of their brain, which is like, really, so what's the rest of your brain doing? Is it just dead weight? Is it extra? Is it a safety mechanism? And there's a number of interesting things about that notion there, especially this, so there's if I only use 10% of my brain, then I have all the other 90% as potential, right, to develop into something else. Which is a problem because you can only state that you only use 10% if you know what 100% use is. What is that? Well, we could discuss that for several hours. There's the other alternative interpretation, which is that, well, we have all this reserve, we have all these resources that we could tap into and that we don't use. And I'm going to come back to later in my talk, considering the amount of energy that powers the functioning of the brain, because one of the prevailing notion is that there's ample energy, much more available to the brain than we actually use. And that will be one of the facts that I'd like to examine. But back to this idea that we only use 10% of the brain, I started looking into where did that possibly come from. Fair warning, the answer is still, we don't really know. It's one of those, we didn't use the word meme at the day, but it's one of these rumors that just spread like wildfire because they're round numbers. They sound nice. They sound hopeful, right? There's all these other things that you can learn and that you can do. But I realized that if you looked at textbooks, this is what they said, that the human brain is made of 100 billion neurons and 10 times as many glial cells. If you squint and do some poor math, that kind of comes up to 10% of the cells of the brain are neurons. So, of course, this is a glia-centric conference, so you're not the people to buy that argument, right? That you only use 10% of your brain cells. But I figured, okay, fine. But by the way, you guys do tend to like this idea that astrocytes are the most common cells in the cell type in the brain. So we'll check that one too. But so here's what I realized, trying to get to the heart of these claims. Where are the data? Who ever said that we have 100 billion neurons in our brain and 10 times as many glial cells, and by the way, that astrocytes are the most common cell type? And these were all statements, mind you, that 20 years ago, you could get by with making them the opening sentence of your paper, and no editor or reviewer would ever ask you for a reference. Everything else, yes, but this you could get away with claiming, which is really surprising because it turns out that there was no actual data, really. If you'd like to dive into the details of that story, how come we got to those numbers, I can recommend to you this review that we published in the Journal of Comparative Neurology a few years ago. But essentially, the answer to those numbers, 100 billion and 10 times as many glial cells, was a combination of taking an order of magnitude estimate, which is a very good order of magnitude estimate, something in the order of 10 to the 11th power neurons in the human brain, and a game of telephone with the actual number of glial cells that you do find 10 glial cells for every neuron in some very restricted structures in the brain, but somehow that seemed to get extended to the brain as a whole. And that lived on. No data, but you could get by with these statements and actually put them in very highly regarded textbooks like Condell, Jessel, and Schwartz. So how come we were stuck there? I think it was a combination of two things. One, that no one seemed to bother checking simply because everybody thought that everybody already knew that those were the numbers, and if you didn't have the references, that was just a minor inconvenience on your side, that if everybody knew already, then that's fine. Life goes on, let's study other things. And I think the second component was that determining how many neurons and how many glial cells you have in any brain, much less the large human brain, seemed to be way too much trouble, given that the technique available at the time was stereology. And stereology consists of trying your best to do to use unbiased sampling schemes, but that are still, however, going to be sampling schemes from a few regions of interest to then come up with an estimate for the whole. And you see in this image here how problematic that is for trying to come up with numbers of neurons or any other cell type for the whole brain, because the distribution of neurons in the brain is extremely heterogeneous. So exactly where you place your samples will have an enormous impact on your estimates, on your results. By this time, I was an assistant professor at the Federal University in Rio, hired to work on science communication, but at my hiring interview, they told me, we know that you have a research background, so if you do become interested in doing science again, we'll have your back, we'll strongly support you. So I realized that, and the fun part of that story was that my main collaborator at that time was Roberto Lent, who was just in the process of writing a textbook called 100 Billion Neurons. So I walked up to him and said, hey, where does that number come from? And he was like, well, everybody knows that that's the number. Why, is that not the number? And I told him, well, what if it isn't? But I think I know a way to figure that out. And hear me out, it's gonna be by turning brains into soup. Because if the problem is the heterogeneity, if the problem for counting cells is the heterogeneity of the tissue, then let's literally dissolve away that heterogeneity. Because if we manage to dissolve the entirety of the brain into a soup that contains free nuclei, then as long as every cell has one and only one cell nucleus, by counting cell nuclei, we're actually counting cells. And the beauty of turning up the brain or the structure of interest into soup is that now every single sample, every single aliquot is representative of the whole. So this is how it goes. You start out with any dissectable tissue, any dissectable part of the brain that you're interested in. You have to fix it very well in paraformaldehyde so that the nuclear membrane becomes resistant, becomes mechanically resistant to everything that comes after. Because what comes after is you're gonna dissolve that tissue in detergent, in a glass tube where you literally grind the tissue between two glass walls. But what that does is it dissolves the cell membranes while preserving the nuclear membranes, which remain intact, because the nuclear membrane is actually the membrane of the endoplasmic reticulum, which is chock full of proteins, and with the fixation, those become so interlocked, so tightly interlocked, that I've tried 20% SDS by mistake. I've tried sonicating these nuclei to get to run controls for how much DNA I could possibly be missing, and I could not destroy the fixed nuclei. So that's very handy, because once you have these fixed nuclei in suspension, you can just swirl or agitate your suspension, make the distribution of those nuclei even in the suspension, and now you can collect samples. And any small aliquot, like I said, is representative of the whole. If it's not representative, you know right away, because there's going to be variation from one aliquot to the other under the microscope. And so this is what the free nuclei look like. You just stain them for DAPI. And so this gives you, in 10 minutes under a microscope, an estimate of cell nuclear density in that suspension, that all you have to do next is multiply by the volume of the suspension, which is a volume of your choosing. You just round it up to any volume that is easy to work with. And in 10 minutes, you have an estimate of the total number of nuclei, and therefore the total number of cells that you had in that suspension of origin. Now, the next thing is, of course, we want to know how many of those cells, how many of those nuclei belong to the different cell types. And nu-N is our marker of choice, because for our ends and purposes, we can consider that nu-N is expressed in every single nucleus of every single neuron that we're counting. Of course, it's not. There are small populations of neurons that do not express it, like Purkinje cells, for instance. But those are, when you're counting, when your goal is estimating total numbers and proportions, that is, nu-N is actually a very good and useful marker. And that also means, I mean, I can go back and forth here, and you see how easy it is to quantify the nuclei that express a particular marker. This also means, because there's no background here, that means that you can use any marker of your choice. It can even be a cytoplasmic marker, as long as some of it is also expressed in the nucleus, is also found in the nucleus. You can use any marker that is found in the nucleus of a particular cell type to go back to these samples and determine, okay, how many cells of that type do I have? So let me give you a quick overview of what we found. And this is, everything that's coming next is work that was possible because of network of collaborators who were willing to provide me with brains or brain hemispheres from their freezers that I could turn into soup. You can imagine how difficult it is to part with an elephant brain that was super, super hard for you to collect. But anyway, there was there was some cajoling and some explanation involved, but at this point we've been able to collect data on over 70 mammalian species, and through collaborators upwards of 250 different species now that span the wide evolutionary tree, so we can really talk about diversity and rules and diversity that define what brains are made of, and that go and actually talk about evolution instead of just comparing mouse brains to human brains and saying, haha, whatever is different means that the human brain evolved to be a certain way. Okay, so what do we find? We find that if you look at each point is one species, and you're looking at the number of neurons as a function of brain mass. And on the right, you're looking at the total number of non-neuronal cells, so every single cell in the brain that is not a neuron. And of course, that encompasses different cell types, endothelial cells, microglial cells, and the actual glial cells, which are astrocytes and oligodendrocytes. But you can see how these distributions are different. If you take any warm-blooded vertebrate brain, the number of non-neuronal cells that you find in that brain is a simple proportionality of how large that brain is. Not so in the, when you look at the numbers of neurons. And these data seem fairly compact simply because we're actually looking at a distribution that spans 5 orders of magnitude. But what you see here, if you look closely, is that for animals of a similar brain mass can have numbers of neurons composing their brains, but that differ by 5 to 10-fold. And you actually find that there are different patterns here. So there's a primate way, that's the data points in red. There's a primate way of putting neurons in the brain. There is a songbird way of putting neurons in the brain. That's these black symbols here. There's an artrodactyl way of putting neurons in the brain. So this is the whole brain. These distinctions become even clearer when you break the whole brain into the different structures here. And you see that, so this is how many neurons go into a cerebellum, these data points here, in a cerebral cortex, the circles, or in the rest of the brain of different species. And you see that there are very different relationships. There are very different ways to put neurons into different structures of the brain across species. But these, on the right, you're looking at how many non-neuronal cells go into making those exact structures in those exact species. And you see that there truly seems to be something universal going on here. So this is number of cells plotted as a function of the mass of the structure. Divide one thing by the other, and you get the density of these cells. And so you see that neurons vary enormously in density across species with brains of different sizes, with different numbers of neurons. And you see that there are trends that don't apply to every single mammal. I've removed the birds here so you can see you can look at the primates, the red points here. This is the cerebral cortex of primates. And you see that there is a trend that the more neurons that you have in a cerebral cortex of a mammalian species, the lower the density of those neurons, whereas the more neurons you have or other cells that you have, or the larger the brain structure is, the density of the other cells that compose that tissue pretty much stays put. So densities of non-neuronal cells are similar, whereas in larger brains, unless they're primate brains, you tend to find that the density of neurons goes down. What you can infer from that neuronal density going down, given that the density of the other cells pretty much stays put, that wherever you see lower neuronal densities, you're actually looking at bigger neurons. And by bigger neuron, I don't mean just the cell body. I mean the cell body plus every single fiber, every dendrite and axon that belongs to that neuron. The consequence is that you can look at the ratio between other cells per neuron, a number of fraction that we're going to determine of these other cells are glial cells. And the fraction of glial cells per neuron is a number that has been for over 70 years considered to serve as an indicator of how much metabolic support is provided to every single neuron. And so legend had it that the bigger a brain is, the more other cells, the more glial cells you find supporting every neuron. And now that we had numbers, you can see here that that's not true at all. There's an enormous jitter here in how many cells, how many non-neuronal cells you find in forming what I like to call now the neuronal entourage, how many non-neuronal cells you find accompanying every neuron as structures become bigger, and there's no correlation there. The true and very strict correlation that you find is between that ratio and the simple density of neurons in that tissue. And the logic here is very simple, that given that the density of the non-neuronal cells in the tissue doesn't really vary, but the density of the neurons in the tissue does vary, the ratio between the two is the ratio between the densities, which depends simply on the density of neurons. In visual terms, what we have is what's represented here in this little diagram, where the neurons are the orange cells in front of this carpet of non-neuronal cells. And what you have indicated here is that it doesn't matter what part of the brain of what species you're looking at, the density of the non-neuronal cells is always the same, about the same, but the neurons have very different sizes. So the visual shows you how the proportion between the two cell types will vary as a simple mechanical, if you will, consequence of how large the neurons are. So this brings us to a number of important questions. The first one is, how come only neurons vary so much in size? And I'll address that at the end of my talk. How do these non-neuronal cells are divided, how are they distributed between the several non-neuronal cell types? So these constant densities that we find, do they apply to every single type of non-neuronal cells? And what are the functional consequences of that distribution? More importantly, is the distribution of non-neuronal cells that you find demand-based, as you would expect if astrocytes or oligodendrocytes or capillary cells were put into brain tissue as needed in order to provide neurons with the energy that they demand. So we could do that in two ways that I'm going to tell you about. One is going back to the soup, because we keep, unfortunately not for the earliest rodent experiments, but for pretty much every brain that we've turned into soup after those first ones, we started storing the aliquots of that brain soup in my freezers, so we can go back to them and just stain them using different markers. And the markers I'll be telling you about that we've been using are IBA1 for microglial cells and S100B for astrocytes. And we did the necessary controls to make sure that they were expressed, these markers were expressed in sections of the different species. This is work that was done by Sandra dos Santos when she was a postdoc in the lab, and Lauren Glassburn, who's now a PhD student at Minnesota. So what did we find? Let's start with the plain facts. Astrocytes are not the most numerous, the most common cells in the brain. I indicated here, these dotted lines indicate 50%. So, and you're looking at the percent of all cells that are astrocytes as a function of the percent of all cells that are neurons. So you find that neurons in the whole brain are, in these species at least, they're close to 50%. The astrocytes are about 10%. And you can break that down into the different areas, and you see that the astrocytes are typically only about 10 to 20% of all the cells in the brain. We can look at how the density of astrocytes varies as a function of the size of the structures in different brains. This is here for just to remind you this is what happens to the density of neurons that's widely variable. So neurons are widely variable in size. And you do see that these are the same species, the same scale on the y-axis, and you see that there is variation in the density of astrocytes, but it's much less than you find for the variation in neuronal density. And also, it looks like the artrodactyls here in pink and the elephant in light blue. They have their own thing going on, but for the other species, including primates that we have, you find that yes, the larger a structure is, the slightly lower is the density of astrocytes that you find in that tissue. And plotting that as a function of the density of neurons, you see that across structures, the density of neurons varies enormously, varies by 1,000-fold. The density of the astrocytes does vary, but only by 10-fold. Does this mean that these astrocytes here with lower densities are bigger than others? Not necessarily, and that is not something that I can answer right now. If we look at the ratio of how many astrocytes you find per neuron, again, you see that there's no universal trend as a function of the size of the structure. But if you look at the more the neurons you find in a structure, so the higher the neuronal density, there to the right, the higher the neuronal density, the fewer the astrocytes that you find serving, if you will, each neuron. And look at that for the cerebral cortex alone, and you have the same relationship where in cortices where neurons are bigger, so with lower neuronal densities, you have more astrocytes per neuron. So larger neurons do come with more astrocytes, but again, simply because that can be explained simply because of the enormous variation in neuronal density given a very small variation in the density of the astrocytes. Something that I'd like to point out, you have to struggle to find the human data point in this relationship here, which, and that's because we're just one more species in the data set. And you see this coming again and again. I indicate the human data point. Humans do not stand out in any variable that we've looked at so far. We are just one more species. We're just one more primate species, and even also in what regards how many astrocytes we have in our cortex, how many astrocytes we have in our brain, how many astrocytes per neuron. Once you're truly comparing apples to apples, once you find that universal relationship to look at, to compare humans to other species, then you realize that our data point is right where you would expect it to be. You also realize here, so now we're coming to the interesting facts, that unless you're a very large brained artrodactyl or elephant with gigantic neurons, so with very low neuronal densities, there's typically less than one astrocyte per neuron on average in your cerebral cortex. So considering what we know now about the many roles of astrocytes in allowing neurons to do their job, we know how important astrocytes is for funding neuronal activity. You could expect then that the distribution of astrocytes should be demand-based. So where you have more neurons, you should have more astrocytes serving them, which is exactly the same question that you can ask of the endothelial cells that form the capillaries that supply brain tissue, therefore supply neurons with the blood that brings the oxygen and glucose that are believed to be distributed and used on demand. And actually, it's very interesting to think of the distribution of astrocytes and capillaries side by side, hand in hand, because we do know that these cells work together. The astrocytes form the capillary end feet that transfer glucose from the capillary blood into the astrocytes, and from there you get the glucose redistributed. So let me give you a visual for what things should look like in a demand-based system, if brains were a demand-based economy where capillaries were distributed at the service of neurons or astrocytes accompanied. So this is a map of the US, and you're looking at the distribution of people. People in a country are what neurons are believed to be in the brain, the units that guzzle energy. So a distribution system that brought resources on a demand-based should look like the system of highways in the US that is very obviously built to serve the areas of concentration of energy use. Where you have more people, you expect to find a denser distribution network. So, which is the opposite of a supply-based distribution system where, like the hydrographical basin of the US, this is all the rivers that distribute water in the country. They could not care less about where the people are. They are where they are, and the people have to just make do with what they get. So the question then was, if we look at the distribution of capillaries in the brain and the distribution of the endothelial cells that make the capillaries, are we going to find that there are denser capillaries and endothelial cells in those locations where we have more neurons, like the highway system? Or are we going to find that the capillary bed is what it is, like the hydrographic basins in a country? So that was the work of Lisa Ventura Antunes as a graduate student in the lab. And what I'm going to show you now is the result of painstaking manual counting of cells labeled as neurons, as endothelial-associated cells, or as the other cells in the tissue. And I think the answer is to the question of, are endothelial cells distributed in the way that you would expect is to serve neurons on demand? The answer is very clearly no. This is neuronal densities across sites in the entire brain of 5 different mice and in 3 rats. And you see at the same scale the density of the endothelial cells across these sites. Very important, there is some variation in endothelial cell density, and I'm not going to show it to you, but this small variation corresponds precisely in a linear fashion with the rate of energy use that you find at rest in the rat brain, across structures in the rat brain. So the distribution of capillary cells it is what it is. Capillaries could not care less about where the neurons are more concentrated in the brain. What happens then to the ratio between capillaries and neurons is something really important, because capillaries are, the capillary cells are the units that bring energy to the neurons, that supply the neurons with energy. So the higher this ratio of endothelial cells per neurons, the higher the rate at which energy is supplied to neurons in one part of the brain versus other, and that's what you find. Again, so this is reminiscent of the, actually this is the endothelial cell version of that graph that I showed you before, where the ratio of the number of non-neuronal cells that you find per neuron varies across structures and across mammalian species as a simple function of how many neurons do you find in that location. So in this case, we can translate that as the bigger a neuron are, the more endothelial cells that serve it. So this is mouse and rat across multiple structures. Lisa expanded her work to the cerebral cortex only of a number of different species, and found that, so the species are indicated here. So we have from tiny little bats to the elephant cortex, sampling the various mammalian clades that we have in our freezers. And you find, first thing that you notice is that there's an enormous range of variation in neuronal density in the cortex of these species, both within, but more importantly across the range of species. And yet, the density of endothelial cells forming capillaries, it remains fairly constant. The consequence of that is that across species, wherever you have more neurons sharing a similar density of endothelial cells, you have a lower ratio, which means that you necessarily have less energy being supplied per neuron. So the answer then is that this is what neuroscientists expected to find, that the distribution of the supply network, the capillary network, should mimic the distribution of neurons, like the highways built to serve cities. What we actually find is something like this, where the capillary bed is what it is, and neurons are served as they use whatever they get. If you have more neurons, they have to share the resources that they have. So, all right, so that's the capillary cells. Now, I want to move on to the last part of my talk, which is glial cells, please. That's what we're here for, right? We want to hear about the astrocytes and the oligodendrocytes. So something very interesting that I'll come back to at the end is that it had been, so studies in human brain, in human forebrain in particular, had estimated that the endothelial cells make about 30% of the non-neuronal cells, giving you a ratio of 2 glial cells for every endothelial cell. So that was for the human brain, and this is the same data the same species that I showed you before. Now just looking at what percentage of all the non-neuronal cells are endothelial cells, and that's data that we have directly from the brain sections, and you see that that 30% is about right throughout. So we can go back now to our visual for our prediction and ask, well, what about astrocytes? Do we find more astrocytes where you have more neurons? So let's start with just those numbers again. What you're looking at is that what fraction of all cells are astrocytes, and this is cerebral cortex only from now on. And so astrocytes are about 10% of all the cells in the cerebral, in the cortical gray matter, they're a bit higher percentage of all the cells in the white matter. And if we now look at only the non-neuronal cells, then what we find is that the astrocytes are still not the majority of all the non-neuronal cells in the brain. What we have is between 10 to 30% of all the non-neuronal cells in the cortex are astrocytes. At the bottom here, I'm showing you data that we published a couple of years ago for microglia, so you can see you have them as a comparison. So if yeah.
So if you don't have large percentages of neurons, nor glial, nor...
I don't have large percentages of what? Neurons, yeah.
Yeah, neurons. So what is the brain component in the case of an elephant? The percentages do not add to 100%.
Because I'm not showing you all the cell types yet. I'm showing you neurons. I'm showing you astrocytes. Remember that this is the percentage of all the non-neuronal cells, right? So I'm showing you that within the non-neuronal cells, remember 30% are endothelial cells, about 7% are microglial cells, which gives you about 10 to 20% of, in the elephant is actually a larger percentage of astrocytes. So you can surmise that the rest are going to be oligodendrocytes of different stages. So I'll come back to that. If you just look at the densities of astrocytes, you see that there's a lot of overlap across the different types of species. But what we really care about is, does that have anything to do with the distribution of neurons and with the distribution of capillaries? So what we find is that in those across species and sites, wherever you have those small variations in the density, in the total density of non-neuronal cells composing the tissue, you have a higher density of astrocytes, which indicates that astrocytes seem to be a somewhat constant fraction of all the non-neuronal cells really in the tissue. Nothing to do with the distribution, with any variations in the distribution of microglial cells. You do have some relationship with local densities of neurons, but first thing, you see that the variation in the density of astrocytes is very small compared to the variation of densities of neurons. And most importantly, you don't have any kind of universal relationship. Where you do have a universal relationship is between actually not even looking at that how many astrocytes you have per neuron. Not even that is truly a universal function of how many neurons you have in that part of cortex, because you see here that the elephant and the various artiodactyls that we have in the sample, they're doing their own thing. They have, so important take home here is the distribution of astrocytes is not universal across every single mammalian species. That's what you get if you look at the ratio of astrocytes per neuron as a function of that local density of other cells. Really no variation there. What you do get is this universal variation of how many astrocytes you find per neuron as a function of how many other cells you find per neuron. And remember that 30% of those other cells per neuron are capillaries. So the hypothesis here that I want to conclude with is that maybe what all of this is explained quite simply if the density of astrocytes that populates a structure is something that gets determined together with the small variations in capillary density that you have in that structure. So let's look at those directly. And here you have this is back to our section by section analysis where we can now look at how many endothelial, so within that non-neuronal cell population, how many endothelial cells do you find per neuron, and how many glial cells, astrocytes, oligodendrocytes, and microglial cells, how many glial cells do you find per neuron? So what you see is that these two things go tightly together. The fewer neurons you have in a region, the more endothelial cells serve every neuron, and the more glial cells come together with that. And you see here that this ratio of glial cells per endothelial cell is actually surprisingly constant. And this is what, if you consider that everything that I've been telling you about has to do with diversity, right? With all these different species, with different brains of different sizes, different numbers of neurons, different densities, and yet, this is something that I find truly, truly striking, that it doesn't matter what cortex you're looking at, there are 2 glial cells per endothelial cells in that cortex. It doesn't matter where, it doesn't matter what species. So there seems to be something very fundamental in what determines how many astrocytes, how many oligodendrocytes go into each structure. So we don't have direct markers for oligodendrocytes yet, and we're not happy with any of the traditional ones that we've looked at. I think the answer is simple. Oligodendrocytes are actually very diverse in including at what point of their developmental program oligodendrocytes are in any part of a brain. But we can look at them by subtraction, and what you have, so to answer your question, when you look at what percentage of oligodendrocytes we can estimate by what percentage of all non-neuronal cells we can estimate by subtraction to be oligodendrocytes, you see that there is this very, very clear trade-off between what types of glial cells you find in the tissue. The more astrocytes you find, the fewer oligodendrocytes you find in that tissue, given this apparent limitation implemented somehow, I don't have any explanation for that yet, that there are 2 glial cells for every capillary cell in the tissue, and that's that. So I'm still working on what's the best visual to show this, but what I'm indicating here is that in any unit volume of brain, you can find a very variable number of neurons, but you always find a certain number of non-neuronal cells. And amongst those non-neuronal cells, 1/3 of them are endothelial cells, about 7 to 8% are microglia, and the remaining circa 60% are divided between oligodendrocytes and astrocytes in this manner that I indicated here. So how could this possibly work? I'm going to leave you with just one more schematic, which is that if you're looking at early in development where you have neuronal progenitors in a certain, in a volume of tissue that has a certain density of capillaries, and we know that at certain early in postnatal development, the brain no longer forms new capillaries. So all that can happen and all that will happen as neurons expand, as neurons grow, and neurons grow, they change their size by expanding dendrites and axons. What happens is that those capillaries get pushed apart, and we know that there's something called the Krog radius, which is the maximum distance that you can have between 2 capillaries. More than that, and you have a dead zone in between the 2 capillaries where oxygen doesn't diffuse in fast enough there to support life, to support the cells. So what we're proposing is that neurons expand in any brain into their capillary bed. They grow their cells grow to the limit of of the expansion of that capillary bed. Once you reach that limit, you're done. And the variation between sites in the brain or across species comes from variations in how many neurons are generated in different sites. How many neurons you have at that point when you're no longer generating new capillaries, because if the capillary bed has a similar if the capillary bed always expands to the limit that gives you that constant density that we found, then you end up finding that in the adult tissue, you have a constant capillary density that is shared, that supports, that supplies a highly variable density of neurons. Now, it is as this forms that you have astrocytes come in first into the tissue, contributing to the expansion of the tissue. And once those astrocytes are mostly in place, now you have the oligodendrocytes coming into the tissue, and you have proliferation happening there. So this is what we will be working on next, is modeling how do you get these distributions that nevertheless always respect the that there's a minimum capillary density that is what supports the tissue, and together with that seems to come a set density of astrocytes and oligodendrocytes, which means that we can go back to that initial question and say that no, there are not more astrocytes where you have more neurons. What you have is just slightly more astrocytes where there are slightly more capillaries, irrespective of the neuronal densities, which means that now that we have the facts straight, we can start to work with, we can start to think about the consequences and the implications for how brains work, for how brains don't work, and how brains evolve, given that you have now we now know this distribution of all the support networks in the brain that, funnily enough, could not care less about the distribution of the neurons. So what we have is this supply-limited economy in brains where neurons must make do with what they have. And I'll end here and thank my collaborators. Doug Rothman at Yale is the person I've been working with in everything that has to do with supply limitation in brain physiology. And Sandra, Lauren, and Lisa are the three talented women who generated all the data that I could show you today. So thank you so much for your attention.